Well, hello and welcome to our first in-person lab of the fall semester for Sal and Malek. This is our fun food dye lab, and a lot of people actually like this lab better than they do a lot of the other ones, actually, because it's colorful, it's easy, and it's kind of fun. Not to say that the others aren't fun, they're all fun. Okay, so what we're doing is testing for the presence of particular molecules in some food dye mixes. For example, Kool-Aid, Mio, various mixes that have pretty colors that have nothing whatever to do with the taste or nutrition of those things. They're there to be attractive to us. Okay, so we're going to consider 6 of the 7 dyes that are available for use in foods by the FDA. We're going to consider yellow 5, yellow 6, red 40, red 3, blue 1, and blue 2. So our job in this lab is to take a unknown food dye of the drink mix and then determine what actual dyes are there that are giving us the colors. Okay, so how are we going to figure this out? All right, the answer is on the back of the food container, but that's not how we're going to do it. Okay, the idea is that these dyes are different colors. They differ in their chemical properties, and one way in which they differ is that they absorb light at particular wavelengths. And we're going to use the spectrophotometer to find out what wavelengths are absorbed by our drink mix. So that's going to give us a pretty good guess right there as to what's in there, because these are very different in terms of their colors. We're also going to exploit the hydrophobicity differences among these, which is going to allow us to physically separate the dyes from each other by reversed phase mini columns. So we'll be able to see the purified dyes separately and then determine the wavelengths for the purified dyes. Okay, so I'm showing you a picture of the spectrophotometer. Now, I think a good way to look at this particular PowerPoint is to get the actual directions for the lab and follow along using the directions from the lab, because it explains certain things that are not there in the directions, and it will help you understand what you're doing while you're doing it. So it goes along with the directions, but it's not explicitly the directions. So the spectrophotometer that we will use to create our absorption spectra, you'll see this in a lab. Okay, and of course, what the spectrophotometer does is it has a light source, and what it's able to do is separate out the various wavelengths of light at our discretion when we set the wavelengths, and that's going to send it through a sample, and the absorption will be measured on a detector. So the different dyes absorb at different wavelengths, and that's because they have different colors, and so we're going to be examining them, and that will be a good clue as to which of those dyes are in our sample. Okay, so this just points out, and I'll show you this again later when it makes a little more sense, that different dyes do have different colors because they absorb at different wavelengths. Okay, so part one is part one of the activities, and we're all going to do that first. And so what we're going to do is observe the color of light as a function of wavelength. So this is to get used to the spec and thinking about what volume of sample you need to put in the spec and how dense the sample can be and still work. And you're going to be looking at the spec, this type of spec. The light path is actually from the back to the front. So a lot of them are side to side, but not this kind. It's going to be, and you'll see that in a second, the light is actually coming toward you as it's going through the sample. All right, so the keyboard things that we will need are the nanometer ones. You adjust the wavelengths up and down with those little knobs, and then we'll be able to set the blank with the knob below, which is the percent transmittance or the absorbance. Okay, so to set the wavelength, you use the nanometers up and down buttons, shown by the arrow in the picture, not the big gigantic arrows beneath the display. Those kind of invite people messing with them, but don't do that, because I don't know what would happen. But anyway, set the wavelength, use the nanometer up and down button. To set the blank on the machine is where you press the zero absorbance button. And you would do that with, for example, a water sample in the light path to basically set the machine for zero. Now, we don't need that for part one of the experiment, but we do need it for parts 2 and 3. Okay, so in part one, we're looking at the colors of light at different wavelengths. And I want to point out that the color you see is not the color it absorbs. If you look on the bottom of the diagram, the blue light is the short wavelength light starting at 400 nanometers. And then the wavelength, as we increase it, goes through the spectrum to 500 nanometers green, then you've got your yellow, and eventually to your red. So the shorter wavelengths are the blue, longer ones are the red. But notice that the food dye color is not the color that you're shining at it. It occurs because the molecule is absorbing that wavelength. So for example, the yellow dye is yellow because it absorbs the blue light, and the red is red because it absorbs the green. So don't let that confuse you. It's a sort of opposite sort of thing. Okay, so I think I just said that. So to our visual system, those are the complementary colors. So the red dye absorbs the green light, so don't get confused by that. Okay, so in terms of using the spec, some of these are getting a little ancient. It takes them a little while to get going sometimes. So when you turn it on, whether by means of the power switch in the back, um, let it warm up. Sometimes it sits there and it looks like it's not doing anything, but it is. And if you open the lid before it's done, it messes up its internal calibration. Now, if it looks like nothing's happening, turn the power off and then turn it back on again. So they all work, but you just have to be patient with them sometimes. Okay, so what we're going to do, we're not measuring any absorbances in this. We're just looking to see what color we see when we are shining the different wavelengths. Okay, so this is a view of the sample holder where the tubes will go. We're not going to use cuvettes. We're just using plain glass disposable tubes. Okay, so this picture shows the arrow on the right is the direction of the light path. And our sample tube will go into the cuvette holder. Okay, so we have set this wavelength to 487 nanometers, and we can see a nice bright spot of blue light that's coming through the machine. So the 487 nanometer light is blue. Similarly, the 546 nanometer is a nice bright green color. And you will record these colors on your worksheet as you change the wavelength in 20 nanometer increments. Okay, here's 630. It's not quite red, but you're getting off to the other end of the spectrum. Okay, so compare your results to the picture. Notice that the color absorbed is not the same as the color that's visible to us. Okay, so part 1B, this is to prepare dilutions of our drink mixes. And we're going to use these for parts 2 and 3. So you're going to make 2 of each one that you're going to use because you're going to need them for 2 different parts. Okay, so the drink mixes, of course, they're concentrates. They're sold like that. And they are, in fact, too concentrated to be read in the spec. Right, you'd max out the machine, it would say 3.5 and flash at you. So you can't actually read the absorbance of the undiluted mixes. So we're going to use 1 to 20 and 1 to 50 dilutions for our experiments. And each person will make duplicate 2 ml sample for one, and then they will choose one and then give the dilution to somebody else in trade for the other dilution. So you're going to make one dilution twice, one for the spec experiment and the other for the reverse phase mini column experiment. Okay, so in terms of how to do this, one way to look at dilutions, which I think is the easiest way, first you decide how much you want to make. And we're going to use 2 ml total volume because that's a good volume to fit in these tubes in this spec. Okay, a 1 to 20 dilution means that 1/20th of the volume of the final solution is the concentrate. So 1/20th of 2 mils is what we're going to use for our dilution. And that's 0.05. 1 divided by 20 is 0.05. So 0.05 times 2 mils equals 0.1 mils equals 100 microliters. So our 1 to 20 dilution, we will get 100 microliters of our dye and add it to water to make it up to the 2 mils. So this is 1900 microliters or 1.90 milliliters of water. Right, same idea for the 1 to 50th. 1/50th is 0.02. So 0.02 of 2 mils is 40 microliters. So the rest is going to be water, and that's 1.96 mil, or 1,960 microliters. Okay, so some people have more experience than others with micropipettes. The big one, the P1000, can only measure up to 1,000 microliters. So you're going to have to pipette twice. Don't try to get the whole 1,960 microliters at once. That might break the pipette. Please don't do that. Okay, so now we get to parts 2 and 3. Oh, I should say, don't forget to vortex the samples. Each person has a vortex. The dye mixes are quite heavy, and so if you just add it, it won't mix well. So we have to make sure we have a nicely mixed solution before we try to do any spectrum on it. Okay, so we're going to blank the machine with water. Oh yeah, parts 2 and 3, we don't want to have too much waiting around for the spec. So you can do either part 2 or 3 first, right, and then switch to relieve the congestion at the spectrophotometers. They both use the dilutions that we just made, and the order does not matter. So pick one that sounds best to you. Okay, so for this, we set the wavelength to 400 nanometers, and we're going to get a glass tube with 2 mils of water in it, and then blank the machine. Then we're going to measure the absorbance of our diluted sample, and then write down the number on your worksheet. Change the wavelength to 420, reblank, and repeat the measurement. Keep doing this until you get to 680. And when we plot all these out on a graph, we will have a spectrum which describes the absorbance properties of our mixture of molecules. Okay, so here's an idealized example. Yours won't look like this. It's not that well drawn either. But Allura Red 40 has an absorbance maximum of 504 nanometers. So if I did this experiment with Allura Red, I would see something like this, maybe a little fatter, not quite as pretty, but you can see that the absorbance maximum is right around the 500 wavelength nanometers. So that would tell me that this is Allura Red among the choices that we have in our solution. Similarly, blue 2 absorbs best at 602 nanometers, and I can see this with this purified dye in that it has a nice peak of absorbance right around 600. So I would identify that as blue 2. Okay, now in our case, we'll be looking at mixtures of dyes, not the purified dyes. Each one has at least 2, and some of them have 3 different dyes. And sometimes they're well separated, but sometimes they're not. And so we're going to have to deal with that when we do our reversed phase experiment. So for example, here's Allura Red and blue 2 together, and their wavelengths are very different from one another, and so they're easily separated by the spectrum. So it's very clear that we have 2 dyes here. One is Allura Red, and the other is blue 2. But you might get something that looks like this. There are 2 dyes in there at least, but they're not well resolved because the wavelengths are too close and too broad sometimes to be resolved by this particular machine. So if we get something that looks like this, we can make a guess or a few guesses, but we might have to rely on some other information to get the complete experiment, to know exactly what's there. We certainly know we don't have blue 1 or blue 2, but we don't know what else. Okay, so here's part 3, separation of dyes by reverse phase chromatography, which sounds pretty fancy, but it's actually really simple. They sell these little cartridges which contain a compound, well, which I'll show you in a second, which absorbs molecules based on their hydrophobicity. So any molecule that's got any hydrophobic character to it at all will be absorbed by this particular column, which doesn't look like a column. I'll get to that. Okay, so we're going to expect that our dyes are going to stick to the column. But what makes this powerful is that they stick with varying degrees of affinity, which depends on how hydrophobic they really are. And so we're going to be using different concentrations of methanol to try to separate them one from the other. Okay, so the white stuff inside is the column matrix, and that binds molecules having hydrophobicity. So we have large nonpolar parts of our dyes, and they will bind, but they bind with different strengths, and that's going to allow us to separate them. Okay, so here's a picture of the solid matrix, the C18 matrix, and it doesn't look that hydrophobic, but if you look at the CH2 repeated 17 times, that's a long, long hydrophobic tail that's stuck to the silicon on this solid matrix. So that's a very hydrophobic molecule, will interact with hydrophobic regions of our dye molecules. Okay, so importantly, you don't have to know these structures at all. I just want to point out that they do have hydrophobic parts, but these differ among them, right? So yellow 5, whenever you see a ring structure, which is all hydrogens and carbons, you know that that's a hydrophobic part of the molecule. So this has some modest hydrophobicity to it, but it's not extremely hydrophobic. Yellow 6, however, is a bit more hydrophobic, having more of those ring structures. Blue 2 looks even more hydrophobic yet, ring, 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 and the same with brilliant blue 1 also. They both look very hydrophobic. So we expect not only will they stick to the column, but that when we start washing it with different amounts of methanol, they will come off with different concentrations depending on what it takes to get them off. Okay, here's Allura red, same idea, erythrocene, etc. Okay, so we need to have some vocabulary to deal with our column. We're going to load the column, we're going to wash the column, we're going to then elute the dyes by adding successively higher concentrations of methanol. We will collect fractions, also called eluates, and those will contain the separated purified dyes. So follow the directions pretty closely on this part so that you know to put the right methanol on there, or else have to start over, which would be terrible. Not really. Okay, so here's what the mini column is going to look like. We've got the little mini column that we bought, and it's going to be attached to a 10 cc syringe that has a plunger. Okay, so here's the separate parts, and when we attach them, we will then be able to put our dye mixes, or our diluted dye mixes, through the column in such a way that the dyes will stick to it, and then we can then switch to the different methanols in order to elute them successively. Okay, here's our tubes for our fractions. These are not cuvettes, they're just tubes, but they work fine in the spec, and moreover, do not have to be washed. Yay. Okay, so here's not exactly our experiment, but very similar to it of what's going to happen. The column matrix is already packed inside, and what we're going to do is basically pour some of our dye mix onto the column, and then rinse it through with a little bit of water to make it stick. So everything is going to stick at that point probably, but sometimes you get a little coming off in the water. So when you start loading, you should actually collect that in some kind of container in case it has some of the dyes in it. But what happens next is stepwise elution with different concentrations of methanol, with the least hydrophobic ones coming off with lower amounts of methanol. And then as you increase the methanol, more dyes will come off until finally everything elutes in the 50%. So this is a diagram of what we're going to do. And we're trying to collect the fractions in different tubes to separate the colors one from the other. And sometimes this can be pretty easy, but sometimes you have to watch carefully and be quick and switch the tube if a different color starts coming out. And if you look at the column during this, you will see the actual dyes on there. And as you start washing it through with the methanol, you will see them physically separate from one another. So take a good look at your column while it's being washed and eluted just to see when the actual dyes are starting to come off. Okay. Warning for this spec, be sure there's enough liquid in it. Remember when you after you've done it, you will notice that the little dot where the light is not all the way on the bottom of where the tube would be, right? It comes through kind of the middle of where the tube would be. So if you have too little in your tube, like the tube on the right, that won't measure on the spec. There's just not enough in there for the light to go through the tube, through the solution in the tube. So you have to be always careful that you have at least 2 mils in your tube in order to get the liquid part such that the spec can read it properly. So pay attention when you're doing this. Watch what's happening and look for the colors in the column. And even if it's still 20% methanol or whatever, if you start getting a second dye out, switch the tube to make sure you separate them as cleanly as possible. As I said before, everybody has at least 2 dyes, but some people have 3. And so resolving those 3 might be a little bit difficult. So you have to kind of pay attention as you're doing this. And that's it. Yay.